I now call to order the Society's 2,455th meeting in the 150th year since its founding on March 13th, 1871. Good evening, everyone.
Good evening. Good evening.
My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends to tonight's lecture by Melissa T.R. Hawkins. I'm delighted that tonight's meeting is being held in person at the Cosmos Club, including a pre-lecture reception and dinner with our speaker. We plan to meet in person regularly going forward and to use Zoom and other social media when it's advantageous in the future. All in-person meetings will adhere to the COVID-related rules of the District of Columbia and the Cosmos Club. Proof of vaccination, including a picture ID, will be required for in-person attendance. Masking is now optional, and we also suggest getting tested for COVID infection within 48 hours before the event. These requirements are subject to change with changes in the COVID-related rules of the District of Columbia and the Cosmos Club. So please check the PSW website often for up-to-date information. We will be live streaming all meetings to YouTube for our remote audience to enjoy and for them to be able to ask questions in the YouTube chat box. And final versions of the recordings will be posted to the PSW website and to the PSW YouTube and PSW Vimeo channels, where they will be available permanently for everyone who wants to view. The Society is grateful to the sponsors of the 2021-2022 lecture series for their support. Policy Studies Organization in cooperation with the American Public University, and a generous sponsor who's asked to remain anonymous. And please join me in thanking them. I am pleased to announce the new members: Caitlin Lucy, Enterprise Strategy Director at the U.S. Pharmacopoeia, interested in biochemistry, microbiology, environmental science, and medicine, who learned of PSW from PSW member Noah Block, who's here tonight. Thank you, Noah. And tonight's speaker, Melissa Hawkins, who learned of PSW from our invitation to her to speak tonight, and whose interest will be clear in some small part from tonight's lecture. Please join me in welcoming them all to membership. If you're not a member and would like to join PSW or support the Society, you can do so easily through the PSW website using the blue Join button on the upper right-hand corner of the home page. We welcome new members and we appreciate donations. Recording Secretary James Hewlin will now read the minutes of the 2,454th meeting and the lecture by Nicole Younger-Halpern on quantum thermodynamics.
Good evening, everyone. On February 25th, 2022, from the Powell Auditorium at the Cosmos Club here in Washington, D.C., President Larry Milstein called the 2,454th meeting of the Society to order at 8:03 p.m. Eastern Standard Time. He welcomed new members, and the recording secretary read the minutes of the previous meeting. President Milstein then introduced the speaker for the evening, Nicole Younger-Halpern, a fellow of the Joint Institute for Quantum Information and Computer Science, and a theoretical physicist at the National Institute of Science and Technology. Her lecture was titled Quantum Steampunk, Victorian Thermodynamics Meets Quantum Computing. Steampunk is a genre of literature, arts, and film, combining romanticized 19th century settings with futuristic technologies. Younger-Halpern said steampunk enthusiasts dream it, while quantum information thermodynamicists live it. Thermodynamics developed in the 19th century as a study of energy, its forms, and the transformations between those forms. The field was motivated by the technologies of its day, such as the steam engine, and it had influence on fundamental physics by asking questions such as, why does time flow in only one direction, and what is matter? Younger Halpern said thermodynamics no longer suffices because modern technologies have given scientists control over extremely small systems, such as single strands of DNA, which thermodynamics alone does not explain. Quantum physics, she said, is the physics of the small. Still, even in small systems, thermodynamic concepts such as heat and work remain relevant. Quantum information theory studies how scientists can use quantum phenomena to process information in ways impossible with classical systems. Quantum phenomena include entanglement, non-commutation, disturbance by measurement, and discreteness of energies. Quantum information theory has been motivated in part by the promise of quantum technologies, and it focuses on how information is stored and processed. Using this toolkit, Younger Halpern said that thermodynamics can be updated to become the field of quantum thermodynamics, which she has dubbed quantum steampunk. Quantum steampunk re-envisions the laws of thermodynamics for small quantum, far from equilibrium, and information processing systems. It asks how non-classical resources can enhance thermodynamic tasks, and which features of thermodynamics are truly non-classical. The speaker then gave a primer on qubits and quantum systems. She said entropies represent uncertainties, and then related through multiple equations how entropy quantifies the maximal efficiency with which some information can be processed. Entropy also exists in statistical mechanics as the spread of probability across possible states. The speaker then said that beyond having conceptual overlaps, thermodynamics and information can help each other. She began by explaining Slayard's engine, which states that information can convert the random energy of heat into focused work. By placing a classical particle in a box and placing in the box a partition connected to a weight, the particle will bump against and move the partition. This movement pulls the weight up and causes it to gain gravitational potential energy. In exchange for this work, information is lost about where in the box the particle is located. This process can be reversed by what is called Landauer erasure. By allowing the weight to pull the partition towards its original position in the box, work is converted into information about the particle's location. In other words, because erasure costs thermodynamic work, computation has a fundamental energy cost, which the speaker said meant that computation is fundamentally related to thermodynamics. She then explained entanglement, which manifests in correlations between measurement outcomes. Younger Halpern illustrated the concept by describing a whole system measurement to accurately predict the states of two connected qubits. She then described a system for erasing qubits entangled with some other system, which she called a memory, that can be used to perform or extract work. Burning the correlations between the system and the memory is fuel for turning information into a resource. Quantum thermal machines are a subfield of quantum thermodynamics. One such machine is the quantum many-body engine, based on the phenomenon of many-body localization, or MBL, a phase of quantum matter. Younger Halpern explained the math for how MBL works. If the disorder is strong compared to the frequency of which qubits change state, the system will appear frozen. This behavior sharply contrasts with particle equilibration, in which cold, clumped particles will eventually move to fit the size of their container. This comparison shows that MBL behaves athermally. Athermal engines can serve as resources in thermodynamic tasks. In that way, MBL can be used to perform thermodynamic tasks, to wit Younger Halpern's MBL mobile operating on an Otto cycle. She explained with graphs and equations how MBL engines can be scaled up to the thermodynamic limit. She said advantages of using an MBL mobile are robust scaling, high power density, reliability, and relatively few worst-case trials. The speaker then concluded her lecture by forecasting that experimental realizations, optimizations, and thermodynamic applications of MBL in the future. Rather, that those things are in the future. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. He adjourned the meeting at 9:59 p.m. The temperature in Washington, D.C., 7 degrees Celsius. The weather, partly cloudy. And the number of people attending in person and concurrently online, 75. Views on the PSW Science and YouTube channels, the PSW Science YouTube and Vimeo channels in the first 2 weeks of the lecture, 299. Respectfully submitted, James Heelan, Recording Secretary.
Now, Okay, are there any comments on the minutes? Online members can submit comments to corresponding secretary Robin Taylor at correspondingsec@pswscience.org. Is there a motion by a member to accept the minutes as read? And I have a second. All members in favor? All members opposed? The minutes are approved unanimously and will be posted to the website in due course, subject to any corrections necessitated by comments submitted electronically. And we now turn to tonight's speaker, Melissa Hawkins, and her lecture on how museum collections inform taxonomy, systematics, evolution, and biodiversity. Melissa is research zoologist and curator of mammals at the National Museum of Natural History. And concurrently, she is a research affiliate in the Department of Biology at George Mason University. Previously, she was a research associate in the Smithsonian Conservation Biology Institute at the National Zoo, and before that, curator and faculty member at Humboldt State University. Melissa's research utilizes sequence information from ancient DNA, that is DNA extracted from museum samples, to study speciation, biodiversity, and evolution. The DNA in these samples typically is degraded by preservation circumstances and processes that occur during storage. Melissa combines studies of museum specimens with field studies, and she has led 2 field expeditions to study mammals in Kinabalu Park in Sabah, Malaysia. And she has been particularly interested, I believe, in squirrels and lemurs. Melissa is an author on numerous research publications and 2 chapters in the Handbook of Mammals of the World. Among other honors and awards, she is the recipient of the first Smithsonian Conservation Biology Institute Emerging Scientist Award, and she was awarded the Best Dissertation Award from the Department of Environmental Science and Policy at George Mason University. She earned a BS and an MS at Western Illinois University and a PhD in Environmental Science and Policy at George Mason University. All questions will be fielded in the Q&A session following the lecture. Melissa, the stage is yours.
Well, first of all, thank you all for inviting me to come here tonight. It's been a privilege to get to speak and talk about some of the research I've done. Most of this research was started during my dissertation and some from my postdoc, and then I'm going to end with a little tidbit of what I'm doing now at the Natural History Museum. And it might seem a little short on the Natural History Museum portion. Many of the chapters or projects are currently being continued there, but I joined the Natural History Museum in January 2020. So what happened in March of 2020? So my time of curation in person has been rather limited, and I've been mostly a virtual curator. That is now starting to change, so a lot more projects will be coming out of the things we're working on right now. Okay, so I hope to convince you all of how awesome and exciting and relevant museums are and how important they are for things like taxonomy, systematic, and studying biodiversity. And first, I had a nice introduction, but a little bit more background on myself is, first and foremost, I consider myself a mammologist. I've been studying mammals since 2005. I'm often told I look so happy in pictures like this, where I'm holding some of my favorite little creatures like this eastern chipmunk. I am interested in mammals because, one, they are more diverse than people think. There is a lot more discovery to be had in mammals. Two, they live in almost every region of the world. They live in Antarctica to the North Pole. They live in the deep oceans. They live in the deserts. And they can adapt to be almost anywhere. They also range in size more than any animal, from the smallest being about 2 grams to the blue whale, which is the largest animal that has ever lived and spans about 6 body size order of magnitude. I love museums. This is a picture from the whale warehouse we have out at the Museum Support Center. If you can't tell very well, this is one whale skull looking from the bottom up. This is a blue whale, and these are real adult humans that are not particularly short-statured. So you can see that curation of mammals comes in a wide variety of challenges, from things like storing blue whales to things like storing shrews. So we kind of have challenges there. I'm also interested in diversity. I always put this slide in because it's some of the weird mammals, right? We have like sloths and armadillos, and I think highlighting the diversity in mammals is super important, and understanding where they're found and why they are found in those places. And finally, I like to study evolutionary biology or systematics. And this is the relationship of how animals are related to one another. Please do not try to read this tree. It is actually a tree of bacteria. It is a pretty tree, though. So that's just to highlight the point that I like to study the relationships of these animals. So I'm going to focus on two major study systems, and the first of which is Southeast Asia, where I worked for my dissertation and doctoral research, and just a couple pretty pictures that are not mine of beautiful, wonderful animals from Southeast Asia. Things like the colugo, the world's most membrane-enclosed animal, more so than a flying squirrel because their tails are enclosed in membranes. So next time you need a weird factoid to tell people, there's one for you. Also to things very charismatic like gibbons and clouded leopards, and also some of the very cool plants like the Rafflesia, a weird vine that flowers and emits a kind of rotten flesh smell to attract flies to pollinate it. So a very cool life history. And then the other study system we'll talk about later in my talk is based in Madagascar, with things like the eye, the fossa, a leaf-tailed gecko that you could ask my husband when a guide pointed one to us. I had to stare at a tree for about 15 minutes while they're both like, it's literally right in front of you. These very cryptic animals that have extraordinary adaptations to blend into the leaves that they are, or branches that they are laying upon. And then, of course, the iconic baobab trees. Okay, so there's a lot of threats to both of these areas that I work in, and I've put up some graphics of forest cover changes through time, which we all know are very sad and depressing. We won't linger on this, don't worry, but as you can see, Borneo has gone from almost a nearly forested island to less than half of it forested today. Madagascar from 1950s to 2005 doesn't look maybe quite as dramatic, but there was a lot of degradation by 1950s in Madagascar. The major anthropogenic force of destruction in Borneo is terracing for palm oil. You can see these patterns here is where they've clear-cut forest and planted palm oil plants for agriculture. And then in Madagascar, they do what's called slash and burn agriculture, or tavi in the local language. And it's such a problem because part of the cultural significance of tavi in Madagascar is if you burn the land, you own it. So there's a lot of problems with land ownership in Madagascar, and it causes people to want to burn down the forest so that they own more land, which obviously you can see the problem there. So I have put up a couple of different focal research questions that I like to study. I'm very interested in adaptive radiations of mammals. I think adaptive radiations, most people automatically think of Galapagos finches, and I personally think that's one of the most boring examples of adaptive radiation. So I'm going to try to convince you of at least one that's, in my opinion, much more exciting. And yes, it is in a squirrel. I'm also interested in how geology interfaces with evolution and the rates of diversification, and what makes cryptic speciation a thing. We've seen it in a lot of groups, in a lot of types of mammals, but why is it? What drives it? And then also, does genomic speciation correlate with morphological variation? So first, obviously, I am a curator at the National Museum at the Smithsonian Institution. We have the largest collection of mammals in the world, about double the size of the next largest collection. And they're an incredible resource for genomics. This is something I started doing in my dissertation work. And I didn't coin the term museomics, but I will use it here. It was actually coined by the thylacine sequencing project. And it's essentially using DNA content recovered from traditional museum specimens for large-scale study of nucleic acids. And here is a picture of me at the American Museum sterile sampling skulls. Oh, wow, that's a tough one to say. Sampling specimens in the collections here for what I affectionately call crusties. This is a picture of one said crusty, and this is a piece of material, either muscle or cartilage, that has been stuck to the skull of this animal and stored in a cabinet for oftentimes well over 100 years. So when we process museum specimens, we use things called dermestid beetles, and they clean and eat the flesh off of the bones, but leave the bones intact. Well, sometimes if you take a skull out of the dermestid beetle colony too early, there are these pieces of material left on them. And that's often what I'm looking for. It's the opposite of what the morphologists are looking for. They want a clean, beautiful skull. I want a real dirty one so that I can pull these samples off of them. And from this, I can extract DNA and try to recover signals for reconstructing phylogenies or looking at population genetics. And everything changed because of the technology when the first next generation sequencers came on the market. So here is what the cost was sequencing for megabase. This is the era of Sanger sequencing, traditional Sanger sequencing, and here's this dramatic change and decline in cost. And that's when the first next generation sequencing machine came on the market. And since then, several more have come on. The technology has changed quite a bit, and the cost keeps getting cheaper and cheaper. So the first human genome cost $2.7 billion for a 3.2 gigabase genome. Now I can get a really high quality genome for about $4,000. So that has allowed people like me, who do not have $2.7 billion, to do research. And again, I've already told you why museums are really incredible, but we have taxa from animals all around the world in the cabinets downtown a few miles from here. So I can open a cabinet of bats and be transported to Eastern Africa. I can turn around and find some squirrels from Southeast Asia, and then I can go and actually open a cabinet of thylacines that we have, an extinct animal. These animals can be impossible to study if they're extinct, 2, if they're really difficult to catch, and 3, if you have projects like mine that have very lofty goals and often span about 12 different political jurisdictions, you can imagine trying to go in the field and get samples collected in 12 different countries in Southeast Asia would take an entire career and not the length of a dissertation. So I use these specimens kind of as a time machine so that I can get DNA from them while still in my comfy air-conditioned room here at the Smithsonian. I'll talk about the field components of my research in a different few slides from now. So the advent of next generation sequencing has really allowed the opening of doors for these projects to be possible. And it also highlights the importance of the collections that we have. So I could have done my entire dissertation without going in the field and been able to have largely the same impact for certain projects and not having brought in any new samples. But the samples are not great because typically when you run an agarose gel of DNA extractions, you want really big, high molecular weight DNA so that you can amplify large portions of the genome in very few pieces. That's the goal, and that's what's shown by this gel here, high molecular weight DNA. Well, this is more of what we get for the museum specimens. This size standard on the side there is about 100 base pairs long. So we have very, very degraded samples because they were not stored to preserve nucleic acids, right? They were stored to preserve the skull in a box for perpetuity. Sorry, Mary Faith, I put the picture I took of us the other day in the lab. This is us, this is me, there's Mary Faith. And we are wearing our Tyvek suits because we're not trying to prevent ourselves from being exposed to something from these specimens. We're trying to keep our cells away from the samples that we're working on. So we don these fun outfits and then we process them in the historic lab, which we call the historic lab at the Natural History Museum. We have a separate ancient DNA lab, and there is a difference between that, and it's generally just based on how bad is your DNA. This is like medium bad DNA that we work with. We don't have extremely rare samples like from a single seed of a piece of corn from an archaeological site that's 150,000 years old. That is a gross overestimate, so don't quote me on any archaeology dates here. But the point is that these are not the oldest, most ancient of DNA, so we have a separate space to keep that ancient DNA more pure. So a little bit more on museums. In the Western Hemisphere, there's over 5.2 million mammal museum records. Not sure how clear you can see this table, but number one on the list here is us at the USNM, is the Smithsonian National Museum of Natural History, and we have about a little over 600,000 mammal specimens. You can see where they're distributed across the Western Hemisphere, but less than 10% of all these samples are actually associated with high quality tissue samples. And at our museum, less than 3% have tissues. So we have to get really clever on how to get this DNA out effectively and use these historic lab. I'm going to kind of briefly talk about methodology here, and that's going to apply for pretty much all the case studies I'm going to talk about afterwards. So this is one of those next generation high throughput sequencing machines. There's lots of names for them. They all are referring to the same short read sequencing platforms. I have not really gotten into the long read sequencings like MinION or PacBio at this point because they won't work on these degraded samples. So they're not very beneficial for these projects. But generally, I am going to sequence that tiny little piece of crusty that I extract DNA from and getting the complete mitochondrial genome and a type of marker called ultra-conserved elements, as well as we can do complete genomes. So ultra-conserved elements, just to familiarize with you, these are nuclear markers biparentally inherited. And the ultra-conserved part of them is highlighted in this graph. The central part of the graph where you see this UCE core has a high degree of conservation across all of tetrapods. So there's not much difference, which is not very informative when you're doing things like making a tree, right? But as you sequence outside of that, you get more substitutions and DNA mutations. And that's the important part for building phylogenetic trees. So that is what I have used substantially in many projects to look at speciation. Okay. There's a lot of bioinformatics, a field I never thought I would be involved in. I never took a computer science class. I should have, in hindsight. But essentially, instead of generating a couple pages of a novel from Sanger sequencing, you're getting like a dump truck full of sequences with these next generation platforms, but they're short. So you have to put them back together. So you have to say you have 50 words of a book, and then you have to find 25 words that overlap with that, and then 25 more, and put them all back together so that you have something that's informative and readable. So, I'm not going to spend time talking about the ins and outs of bioinformatics, but essentially we use a lot of pretty well-established tools in order to clean and quality check our data, put the sequences back together and get them to a point where we can align multiple individuals and build phylogenetic trees. I'm somewhat of an expert in hybridization capture or enrichment arrays. These are in solution enrichments where we can essentially use published data and published genomes and pick out the exact genes that we want to sequence, put them in a commercial kit based on either RNA or DNA baits, and recover that, enrich other samples onto those baits, and recover the same sequences from many, many different individuals. Or the dumbed down version or simplified version of that is picking the red apples out of your bucket of mixed red and green apples, if you consider a bucket of apples like your genome. So essentially, you can look at the figure on the right, and it shows you a bunch of things of how we do this with magnetic beads and annealing your sequences to the probes that are attached to the beads, and then you pull them apart, and then you can sequence what has been enriched. Or again, you can grab for the red apples that you're targeting. So we have published 2 papers. One is looking at recovering a wide variety of mammalian mitochondrial genomes. And then the other one, paper shown on the bottom right-hand corner here is what we deemed the ectobaits array, where we would enrich simultaneously the host, parasite, and pathogen of a sample. So if you had a tick that you found, you could extract the blood meal of the tick and figure out which species of tick, if the tick had a pathogen, and what it fed on. So that was kind of fun. I have a little tangent here about my first graduate student's research, because she optimized doing standard traditional microsatellite markers from museum specimens, but using next generation sequencing. So we figured out how to do what has been called genotype by synthesis, where you do large scale genotyping instead of Sanger-based genotyping. And we looked at how it varied across museum specimens. So this graph in the green is our tissue control, how it performed at different microsatellite loci, and you see it's all pretty high percentage of confirmed alleles. In the middle is what we had a higher concentration museum specimens, and on the right is lower concentration. So you can see the museum specimens go from working really well to working really poorly. So we kind of had both situations. So you have a lot more dropout here. And this was based on the Humboldt's flying squirrel and my first student, Stella Wan, who published the methods paper with that, as well as the subsequent phylogeography, which just came out a couple months ago. And again, we showed by using these museum specimens that we could recover signatures of a population that's no longer, it's an extirpated population south of, in the San Jacinto Mountains by Los Angeles. So it's kind of interesting that we found that this one little population that's now gone had a unique haplotype and signal, and that can be used for conservation decision-making and inform the status of this species across its range. So if we jump back over to my main focal groups again is in these geographic regions of Sundaland and Wallacea. They're both in Southeast Asia. I'll try again with the pointer. Nope, no pointer. So Sulawesi is on the right-hand side of the orange line on the left side of the screen, and it's kind of a weird K-shaped island. On the left side of the line right across that is Borneo, which we're going to talk a lot about today. And interestingly, these areas, both being hyper-diverse, but they've had a lot of change through time. So on the right-hand side, you can see the sea level. And if I go back about 10,000 years, you could see that the islands become one kind of super island. There's no discrimination between Borneo, Sumatra, Malay Peninsula, and Java. So when these islands were connected, it allowed the movement of animals to go between them. So here's current present-day sea level, and then in the past. So obviously that sort of connectivity of land and thus connectivity of forest can allow animals to cross back and forth across this exposed shelf. So we want to look at the patterns of colonization across these areas. So the first thing we did was a study looking at published data. Look at how long ago did sister species diverge on these different land masses. And what we found, to simplify this project pretty dramatically, is that Borneo was often much more divergent genetically from Malay Peninsula and Sumatra, which makes sense because it's the first to disconnect when the sea level comes back up. But also interesting through the plot on the top, but you can see that this is a change through time. So this wasn't a single time that all the animals moved from Sumatra to Borneo. You could see it's more of a process, and that in the Pleistocene, which is all that's shown here in this diagram, is repeated connectivity and isolation and connection of these land masses, which has kind of served as a process by which speciation has occurred. So if we cross Wallace's line to the island of Sulawesi, this is a paper I published during my PhD looking at the colonization patterns of the squirrels of Sulawesi. I wish that there was either better tourism in Sulawesi or better, more forested areas that you could get better pictures of animals in life. But unfortunately, most of these are either artist renderings or the couple pictures that I can actually validate that are the correct species online. So the top 3 inset maps show the distribution of the 10 named species of squirrels that are found in Sulawesi. And you can see some of them have very strange, tiny ranges, and others are more widespread. And these animals have adapted into 3 genera: a pygmy or tiny little squirrel, a giant land squirrel, and 2 species in the genus Hyoscyrus, which has a really long snout adapted for eating earthworms. So, like I mentioned, there's only 2 pictures that I can actually say are Sulawesi squirrels on the internet. And shown here, one is the pygmy squirrel on the left and Procyrillus leucomus on the right. It has little white patches behind its ears. But the other species are really quite astonishing. Some of them have banding in their tails. Some of them have ear tufts, which is a very squirrely thing to do, is have different color and different length ear tufts. The giant red squirrel is really quite stunning. It's bright red underneath, and its head body is about a foot long, so it's quite a large squirrel. And then the long-nosed squirrels that eat earthworms. So they have kind of filled all the niches and are the only squirrels that have crossed Wallace's line. So we wanted to look at when did these species diversify in this group and what was kind of the driving force behind it. So we're going to back up 20 million years and look at what was Sulawesi 20 million years ago. Sulawesi is very geologically complex, and it's comprised of at least 5 different continental fragments of land masses that have been uplifted and pushed and turned and rotated through time as the plate tectonics have changed. So the light yellow is above sea level, the orange is higher elevation, and then the brown, which you'll eventually see, is above, I believe, 2,000 meters. So if we jump, another 5 million years, you see some of the high elevation areas actually gone away, and then it's back. And then there's other little isolated island chunks above sea level. And then it's only about 3 million years ago that we start to see these high elevation spaces and to what it looks like today. So as you remember from a couple slides ago, there were some species that were really restricted in their distribution to this kind of central area in Sulawesi. And that's probably because there's most high elevation range in that region. So again, based primarily on museum specimens, we constructed this tree from the whole mitochondrial genome, about 16,500 base pairs of DNA. And the first interesting thing we were able to do is date the split of these animals from their relatives in Borneo and other areas. And from this estimate, using mitochondrial DNA, you could see that's about 11 million years ago. So this is a really old adaptive radiation. Again, not to knock on the Galapagos finches too much, but that whole radiation, I believe, is less than 3 million years old. So these are much older animals. And interestingly, highlighted in the green text, you could see one species of the genus Procyrillus is found outside all of the others. So I run to my advisors and I say, I get to describe a new genus of squirrel. This is amazing. Pat me on the back. And my first advisor says, well, don't get too excited. And the other one says, well, you probably have contamination in your data. It's nothing like being a PhD student, right? Well, I didn't have contamination. I had what's called mitochondrial capture, unfortunately. Far less exciting than a new genus of squirrel. When I put the nuclear markers back in, you could see in green again that animal jumps back with the rest of its genus. But what was the animal it hybridized with? Either it's an unsampled specimen, a species that I have not included, or it's an extinct animal, that the mitochondrial signature is just not around anymore. But if you look back at the dating of this tree, you see that now with the nuclear data, the dated cross of Wallace's line is about 12.5 million years. So it's still a very ancient split. And we think that because of all the movement of the pieces of land, that whenever these animals crossed Wallace's line, they hit a land fragment, and then the geology changed and allowed for either this first adaptive release, when you have an open habitat and all these animals could diversify into the extant genera we see today. But also, once new pieces of land emerged and connected, it gave a lot more habitat for animals to potentially go and isolate and speciate further. I will say that the geological reconstructions have changed. A couple years ago, there was this wonderful paper that corresponded perfectly to the dates I've recovered. And then a new paper came out and kind of changed those dates. So the new paper figures are included here, but there used to be, right at 5 million years, like all the land mass is connected. And I said, that's it. That is what caused the speciation within this genus, because all the land became above sea level, and then they could just have all these different habitats to speciate. So maybe another paper will come out and will correspond more perfectly to my data, or I'll have to generate more data and see what happens there. But the takeaway is that it seems really likely that these long-term geological events gave the adaptive release that was needed for these squirrels to start having dramatic changes in both the number of species and the time of divergence between species. So we have, to my knowledge, the oldest adaptive radiation that's currently been studied. This ancient mitochondrial introgression in the pygmy squirrel. Really deep divergence within some species. Some were over 5 million years diverged. That's like generic level divergence, and they're considered sister species in the same genus. We have absolutely no evidence of gene flow in these taxa, which is unlike a lot of the other more recent adaptation or adaptive radiations like Galapagos finches and the African cichlids. And the geology was likely super important in shaping when these speciation events occurred. What would I like to do next with this is I want to have a much finer scale sampling and look at movement and coalescent histories through time. And I would like to do whole genome sequencing for each genus to look at when each genus really split from one another. And we have 134 specimens at the Natural History Museum of most species. The next chapter, so that was a very deep evolutionary history, right? Looking at 12.5 million years. This is a much finer scale, population level project looking at the genetic structure of the mountain tree shrew. And these are fantastic animals. Probably my favorite animal that we trapped in Borneo. One, because they're really easy to catch, which always makes someone like me happy when you're catching animals. But two, because they have like this great personality. I think most people see the movie Ice Age. We've seen Ice Age here, right? And you have that little proto squirrel, right? I think it's a tree shrew. It's not a proto-squirrel. It's got the same dental formula as one of these. It's clearly doesn't have two incisors like a rodent. So I believe that the guy from Tree Shrew, Ice Age is actually a little tree shrew. And these guys are super cool because they live all over the island of Borneo in a high number of species. And this one, the mountain tree shrew, actually has evolved with this pitcher plant, which you see, it's like nature's little toilet here. There's actually been studies looking at the head body length of this animal in comparison to the opening of this Nepenthes loei, this pitcher plant found in the mountains of Borneo. And it secretes a sugar from the top panel where his face is near on this picture, and it acts as a diuretic. So he drinks the sweet sugar from this little Nepenthes, and then it kind of causes him to go to the bathroom right in his little pitcher there. So it's very cool. Other animals also use this, these pitchers, but it's thought that two species of Nepenthes have evolved with this mountain tree shrew. And this is a map on the left of Kinabalu Park, and the little lines, which we'll see again, are the transects we did catching small mammals in 3 different locations. Kinabalu is the tallest mountain in Southeast Asia, and its sister mountain in the back of the park called Mount Tambayukan. It was much less studied, much more intact primary forest, but also much more treacherous because there was no infrastructure at all. So we went out in the field twice, about 2 months each time, tent camping up mountains. So a really good time, but very hard work. And it's resulted in several cool papers so far. So we were there in 2012 and 2013. We sampled from about 350 meters above sea level to over 3,200 meters, where you're at pretty much the edge of the forest, and above that is pretty much sheer rock granite face. This is our base camp at one of the bottom transect around 350 meters, and this was just one picture with some of the gear that we had to drag up mountains. So no easy feat there, and lots of fun times and stories from the field. But here again is our trapping effort across the transects and the elevational plots. And you can see the lines of the elevation on the right-hand side. The mountain tree shrew was our most common animal that we caught, but the most interesting thing about it is it has an extremely wide elevational range. Most animals are like below 500 meters in elevation or above that, and usually with about less than 1,000 meters in elevational span in this system, but this animal was collected 836 to 3,356 meters above sea level. And I'm pretty sure if we went higher and set traps higher, we still would have caught them. They were 37.5% of all of our trapped animals. So again, this is why I like them, because we could get a really robust sampling of them. We collected specimens until we got our limit, which we often hit with this animal because they were so dense. And we would take genetic samples from them if we had reached our limit. So we did standard genetic work. Again, we did UCEs and mitochondrial genomes. One cool thing we did with this is we took the UCEs, which are typically used for phylogenetic reconstruction, and did population genomics with them. So instead of using those variable sites and making a tree, we isolated SNPs from them. And that was new because most people were pulling SNPs from UCEs at the time, but using the SNPs to make trees and not doing true population genetic analyses. So we did all the fun Hardy-Weinberg equilibrium and removed SNPs under selection to look for truly neutral variation. We refined our data to have only one SNP per locus, so we could remove, they would be obviously linked if there was more than one. Ran structure and migrate to look at the patterns of movement across the elevation and between these two mountains where we caught them. we found two really divergent haplogroups in the mitochondrial genome. And if you could see the hash mark there, it's over 180 base pairs different of the sequence between these two haplogroups. So they're really easily to distinguish the two groups. Unfortunately, it wasn't as clean and easy as one mountain had one haplogroup and one mountain had the other, which I'll show you in a minute. There are 36 haplogroups across 83 samples, and when we checked and removed first-order relatives, we still had 34 haplogroups across 54 individuals. So the SNP results, as you can see here, it's kind of this V-shaped plot in the PCA. That's a PCA generated from our complete data set, which has 684 unlinked SNPs with no missing data, which is the nice thing you can do when you have millions of sequencing reads, is you can make sure you have enough data to have a complete data set. And what you can see in the top of this PCA is these blue colors, and those correspond to Mount Kinabalu. And the orange colors correspond to Mount Tambayukan. And I mentioned Kinabalu was the bigger mountain, yet it has much less genetic diversity than Mount Tambayukan. Well, one thing I didn't mention was that Tambayukan is a much older mountain, and it's crumbling. Mount Kinabalu is younger and it's still growing. So they have these two opposing forces here. And what we were able to tell from this is that, oh, well, this species seems like it evolved on Mount Tambayukan and subsequently colonized or kind of rode Mount Kinabalu up while that mountain uplifted. When we ran structure and DAPC, which are two PopGen programs, we got either 2 or 3 genetic clusters as most likely. There's a lot of reasons why there's biases in some of these programs towards 2 clusters. So that's why we show the plots of both here. Interestingly, part of Tambayukan is always distinct. And then if you have 2 clusters, the lower part of Tambayukan and all of Mount Kinabalu are the same. But if you split it into 3 clusters, you can separate Tambayukan from Kinabalu, but having 2 different groups on Tambayukan. So that was unexpected. I once had a talk with my collaborators after one of our Friday seminars. We'd gone out and had fish tacos, and we were drawing on a napkin, and we were trying to figure out, well, if they came from here, then why is there this group here? You know, all these great conversations you have late at night when you're having some margaritas and drawing on napkins. But we were wrong. We were wrong on what we thought. We ran Migrate, a very complex program that models movement across populations. And the Migrate models we tested were 1 through 6, shown on this slide. And of course, probably the most complicated one on this realm of possibilities was found to be the most likely. So that was a bit surprising to us, because these animals are in very small home ranges, and they can live in less than about 150 square meters. Very small home ranges, yet they're having these complex migration patterns where the high elevation and low elevations are kind of having the stepping stone model of movement through their genes necessarily, not necessarily individuals, but through their genes at least. So that was very interesting and unexpected. And despite these steep elevational gradients, we found that there was substantial gene flow, but that Mount Tambayukan really harbored most of the genetic diversity. So what's the explanation here? Why are they able to tolerate so much gene flow, or what's going on? We think it's because this animal is extremely general in what they eat, and they're not necessarily isolated on the high peaks because they use burrows. So maybe you'd think, oh, well, the high elevation, they should evolve a more cold tolerant pelage or something like that. But these animals all use burrows. So they're able to kind of buffer the elements by using burrows. So that's why it's thought that a lot of small mammals kind of violate some of these ecogeographic rules, like Bergman's rule about body size across elevations. The cool thing we did here was really show that we could use these ultra-conserved elements that were initially designed for phylogeny in this really cool shallow time scale, but also for these Sulawesi squirrels, this deep time scale. So I want to, of course, go back to Borneo, and I want to go to other regions of Borneo. You can see the blue stars where we sampled, but I want to get down to all these other areas. And sadly, that still says 2021 that I wanted to go to these places. And obviously that didn't happen. But, maybe I should just, put a 2023 on that slide instead. So I would really love to do some microbiome research and see what kind of specialization they're having across their range as well, because Borneo is a very complex island. So now if we move to Madagascar, this is a fun picture of the colonization of mammals that's known to Madagascar. So this is, I think, the third largest island in the world. It is across the Mozambique Channel from East Africa. And there are a lot of theories about ocean currents that kind of predisposed Madagascar historically to have colonization waves from East Africa. So there are at least 4 independent mammal colonizations, the oldest being the lemurs, somewhere between 45 and 60 million years ago. Then the tenrecs, rodents, and carnivores. And here is, I think, my favorite avian adaptive radiation example. So again, this is like the Sulawesi squirrels I mentioned, where you have one colonizer and then they diversify into a number of different types of bird. And here they have everything from like these nuthatch type birds to fruit specialists and nectar specialists. So Madagascar is really cool to have all these endemic adaptive radiations. It's also split up in a lot of different ways based on literature. So the picture on part A is looking at watersheds and the different major river systems that break up Madagascar. And B looks at current climate zones. And you can see that there's a lot of them, but some areas have pretty large swaths of the same climate, whereas others, like the eastern coast of Madagascar, have very narrow, thin ranges. And that's because on the East Coast, there's a big mountain range. So I was tasked when I started my postdoc with coming up with the largest phylogeny of the group called the Shafaks. They are vertical clinging and leaping specialists. You've probably seen them in a David Attenborough documentary before. They're quite charismatic. If you've gone to the Duke Lemur Center, they have an area you can walk through with the cockerel shafak. And I guess the younger kids than me always say Zubumafu. He's like this cartoon that had a cockerel shafak. And I had no idea what they were talking about until I Googled it. But they are a pretty charismatic group. They're pretty large. They're the second largest lemurs, up to about 6.5 kilos. And what we wanted to do was really generate a nuclear phylogeny, because all that had been generated were very small mitochondrial ones with few samples. So here's the picture of all the living species of shafaks. They're broken in half by the western taxa, which are in that box, and the eastern, and then the strange one, the tattersalls, up in the north. So there are currently these 9 species named, and all the spots you can see on this map are the individual GPS locations of samples we included here. The group I worked with has been in Madagascar for over 20 years and has sampled over 200 different locations in Madagascar. So we had over 200 wild, free-ranging animals included in the study. And we used UCEs again. I made a custom gene set and then sequenced these on an Illumina NextSeq and a HiSeq lane. So here's a tree that shows the major pattern that was expected of the western and eastern species. Again, they're all color-coded to match the tree branches and their pictures. And interestingly, we found hybridization down here between the Decans and crowned species, which is interesting because this is the crowned shafak and the Decans, and they're pretty easy to tell apart physically, but we found a lot of admixture. One caveat is our sample sites were pretty much restricted to this little circle here, right on top of one another. So that is one bias in our data set, and it's our least represented species here. But really interestingly, and this paper has kind of become much more the story about the diadem shafak, that we found 2 divergent clades in this group. And I will keep the colors consistent for the rest of this. And one thing we did is we took our data and we looked at what's called ecological niche modeling, and we projected on a map based on where we had samples what the ecological niche would be. So if you can see in this top part, there is a purple associated with the purple samples in the clade recovered, and blue. And what you can tell when you overlay this climate map is that it almost perfectly corresponds to the current climate zones in Madagascar. Well, this was really compelling, and that was kind of exciting. And I was like, oh, that's pretty clean. Usually ecological data in particular is a lot more messy. So we went a little bit further. We looked at a demographic reconstruction, and although these plots look somewhat similar, the age or the time at which the groups expand through time is different. So showing that back in time, the clade that is purple started increasing its population size a lot longer. It had a longer history of increasing than the more sharp, abrupt increase in the purple clade. So this is just kind of another piece of evidence I'm using to show that this might be distinct between these 2 clades. And beyond looking at that, I had an undergraduate mine through 20 years of field notebook data. And it was from Word documents, not from Excel sheets that are nice and easily sorted. So Philip was a very patient student who plucked all the morphometric data and then generated PCAs. And again, we have 20 years of ecological data. Every time they would dart an animal and put a pit tag, like a microchip like your dog and cat have in it, and they would take morphometrics on these animals. These are living sedated animals, so there's going to be some error. And I really expected no discerning morphological features at all. I expected what would look like a shotgun blast on this PCA for both groups and having nothing discerning between them. But again, we see that especially certain traits had an effect on the data and pulled these 2 clusters somewhat apart. So the blue clade is shown here as being pulled apart by some measurements like its thumb. So that could be something of how they're foraging, how they're moving in the ecosystem. But it was really cool to see any sort of differentiation when you have so many different field collectors putting data in 20 years of data sheets. So this undergrad will be on this paper when we finish it. And the next steps, which again, 2020 came along, and I was supposed to go to the British Museum in May 2020 to work on the type specimen of the species. And the type specimen is what the entire species was based upon. So that's the morphology, the genetic signature, the pelage characteristics are all based from that type specimen. This is a picture of it, the left image here. This is the type from the British Museum. This is one we have in the museum here. And so now I've been trying to get to the British Museum for 2 years now to try to do the morphology on that. And we actually have been approved to get the type sequenced. So we can place the type in these 2 clades and figure out which one it belongs to. And hopefully this year I'll get to the British Museum, do some morphometrics on the skulls, and figure out if we actually have enough morphological characters to say that there are 2 species of diadem shafak. We only have 7 here, and yes, I've already measured them, but they all seem to be from the same population, so I'm not going to be able to tell much without comparison to other collections. One thing that I have been learning in COVID is how to use this really cool surface scanner. It uses sophisticated cameras and computer software to build a 3D model. You could use this to 3D print it if you wanted, but you can also just overlay multiple individuals to look at where there are similarities and differences from landmarks on these skulls. So this is one of the shafaks I have created a scan for here. And the great thing about having this scanner is that it's portable, and I'll take it to the British Museum, scan everything, and then try to figure out if we can morphologically tell apart 2 different cryptic species of Shafaks. So this was the first genomic survey, and we found a couple different things with this possible cryptic diversity in the diadem shafak, as well as admixture between them. So we might need to adjust conservation units to a net zero gain. We might have 2 that are not actually distinct species and 1 that is split in half. So this is still in the works, and we have more to come with that. Okay, and the last thing I'm going to talk about today is the greater bamboo lemur. And we published the genome on this a couple years back. And one thing to note that you might see that there looks to be a dog collar with a tag on this individual. It is not someone's pet. It is a collar for behavioral monitoring. Once it was sedated, they put the little tag on. And this is Jolly. She just happens to have a cute pink heart on her collar. But they know the combination of the color of the collar and the type of tag so that they can go out with binoculars, see the animal, and know who it is. So this study, we have quite a complex situation here. So the greater bamboo lemur is currently found in the tiny little purple specks you see on that distribution map. Interestingly, though, you see the circles of yellow as where there were subfossils. Madagascar has no fossils, but it has a lot of subfossils. So something in the range of 10 to 20,000 years old. The top picture is a subfossil of the greater bamboo lemur. And interestingly, in the northeastern part of Madagascar, you see these green circles, which are museum specimens. So recently, they were up in areas that they are no longer found in anymore. So we did sequencing on them, and one thing I'll quickly note is that we had our samples, we picked as divergent as possible when you have such a small range. We picked a sample from up here in the eastern northern extent of its range in an area called Tortorofutsi. It's a really cool area. We have one from Ranamathan, 2 from Kianjavatu, a long-term study site, and 1 from Karianga. So we wanted to span the distribution that's available as well as elevation of the species. We did 1 high coverage and 4 low coverage genomes with the pretty standard sequencing of Illumina HiSeq and mate pair. And to jump right into our results, we sequenced these genomes, we put them back together, and this figure looks at the demographic history and changes through time. So each major color group corresponds to an individual. The lighter lines are pseudo-replicates, bootstrap replicates, where you take the genome and you split it into 1/100th of the size, a random piece, and then you repeat the analysis. The dark lines are the consensus. So what you see here is the age is listed in readable numbers on top. So about 10 million years ago is back here, up to 100,000 years ago. So you can see there's this gradual increase and that the curves don't match completely between the different sites. And the most interesting thing to take away from this is that all groups gradually increase and then have a really big crash. The crash for the high elevation individuals, the purple and the orange, is much later than the low elevation. So they might have been buffered from the population decline from living in a higher elevation habitat. But all of them have collapsed, about 99% in their effective population size. So we did a little GIS with this one. We made a very coarse minimum convex polygon across the sites where we knew that there were individuals. And then we said, okay, well, let's estimate based on forest cover where these animals used to be. Obviously, it's pretty coarse. We're not saying that they were found in an equal opportunity across the entire range there. But we also added buffer zones to forest because of the known amounts of deforestation. And really interestingly, we had an almost identical estimate of a 97 to 99% range reduction with a 99% loss of their effective population size. So it was pretty interesting that these corresponded so well. And this animal, I should note that there's about 1,000 individuals left. So now if we jump to some of the things that we've been working on here, is optimizing the exact DNA extraction methods to get the best amount of recovery from these museum specimens. This is in process right now, so I do not have anything to say on the results of that. One thing that's a little bit further along is actually extracting RNA from formalin-fixed bat tissues to see if we can find ancient pathogens. This is very important, obviously, of what has shut our world down for the past 2 years. So we're looking at samples right now that were collected in 2002 and 2003 from the northern regions of Myanmar, and they are some of the bats that are potentially suspected. The problem is it's a numbers game. So if we look at wild bat populations and you screen them for coronaviruses, they're about 5% positive. Well, if you're in the field and you catch 100 bats, you can get 5% with an actual number to it, right? But if I'm dissecting a bat in the collection, it takes me, for one, about 30 minutes to do one dissection, and that's one chance to detect it. So right now we haven't had any confirmed SARS-CoV-2, but we have recovered RNA from formalin-fixed museum specimens, which is pretty cool. And we're also hoping to start this year some local small mammal surveys for evidence of endemic SARS-CoV-2 in the DC metro region. You may have heard the stories of white-tailed deer containing COVID, and I'm a strong believer that if it's in the white-tailed deer, it's also in the deer mice. So we want to try to go out, collect samples, which are super critical because at this point in our history, we have a really important opportunity as museum scientists and museum curators to get some of these valuable samples in our collection and archive all kinds of tissues so that we can have some sort of time point and understand the prevalence, what variants are being found in this wildlife, and are there going to be multiple spillovers back and forth? I've heard that there's the first evidence of deer to human transmission. I haven't read that paper, but it seems like, this is not going to be something that goes away anytime soon, unfortunately. But I think as museum curators, we have a really important responsibility to make sure that we can document this and archive tissues the best of our ability, because in 20 years, who knows what kind of technologies we'll have to be able to get the most out of these specimens. So I hope to have convinced you that museums are not these dusty old places that nothing relevant happens anymore, that they are super important for understanding conservation, biogeography, evolution, and that they have more uses every year as technology advances. There is many people that have contributed to all of these projects. I'd like to thank everyone on this slide. Importantly, my two little daughters that are obviously, hopefully sleeping at this point of the evening, and the whole collaborative team that has ranged from my doctoral work till now. And with that, I would be happy to entertain some questions.
Well, thank you so much for a very interesting talk, and it's great to have somebody here to highlight the importance of museums and museum collections, which, having worked at the Museum of Natural History here in DC, are near and dear to my own heart. So I'll take questions from the audience. First question is Carl Merrill here in the front row. If somebody can bring a microphone to him. Carl, keep your hand up.
Of the society. You've covered a tremendous amount of area, and there's all this speciation, and the question is what's driving it. But one thing that worried me was that you're working with animals that many of which carry a number of viruses, particularly the bats. And I know some colleagues who studied that, and they use respirators and other methods to make sure they don't get infected, because it'll get in through the lungs. And as you pointed out, the deer now seem to have it. And by the way, I've got those papers. I only glanced at them, but the ones the one I looked at, they already have sequenced the genome, has 67 or so new mutations in it. So that raises a lot of problems. But the real question is, what precautions do you take about this? And it brings up another interesting problem, because those museum specimens, particularly when you go look at the RNA for viruses and stuff, they're probably as good as ones in the wild. So it means the museums are a little more dangerous than we might have thought before.
Well, I'm not sure that I would think that they're more dangerous. I think we had to work really, really hard just to get the RNA to be about 75 base pairs long. And as soon as we sample from these bats, we are sticking the sample in a buffer that makes pathogens inert. So I'm wearing mask and gloves and a face shield when I do the sampling. And again, these have been stored in ethanol and formalin fixed. So I don't think there's really any chance of getting sick from those specimens. And again, we put them in either, it's called DNA RNA shield or Trizol, which renders any pathogen inert. As for field sampling, we will have to figure out what the best PPE will be to ensure that we're not, getting ourselves exposed. But yeah, we're definitely aware of trying to make sure that it is not jumping into us while doing this kind of research. Most of the people that do the full respirators in the field are in very high density areas. Like if you're going into a cave with bats, you absolutely want to wear a respirator and have the full suit on. Okay.
I think there's a virus in our electronics. It's a virulent form, too. Seems to be communicating. So we have a question over here in the front row. If somebody will bring, we have a blue microphone back there. Sorry. Savannah.
Hi, I'm Savannah Crawford, a member. And so I noticed that for all of your data, like trying to sort data, you used PCA. And I was wondering if that is just the best algorithm that like you have found, or if that's like the industry standard, or if you as like a biologist are interested in finding like better sorting algorithms, because I know that's like an active problem in applied mathematics.
It's pretty standard for genomic data to use PCAs. There are some other figures that don't necessarily look great on slides that we have done. Some are in bar plot form, like I showed. But yeah, PCAs are pretty standard. There's also discriminant function analysis, which works pretty similar to a PCA. If you have other ideas, I'd be happy to hear other ways to get information out of our data.
We have the blue microphone again. If you'll stand up, tell us your name, and if you're a member.
Good evening, Michael. I'm not a member. This is my first week, though I'm sure I'll be joining pretty soon. I have a somewhat less technical question about your museum work, something I've always wondered. I'm sure some of the tests that you run, or most of the tests you run on your samples, are destructive to the samples. So I'm curious how you determine when it's the right time to use a new technology on a sample versus holding that sample for a potential better technology in the future.
That's a great question. Yeah, our goal is to be kind of the advocates to keep the specimen in perpetuity, right? But we also want to get more information out of our samples. So it's something that we have an application for. You'd put in and justify how much you need, what you need it for, what it will show, and a committee evaluates that. Whenever I sample a specimen for the little pieces that I use, oftentimes it's not actually bone. So it's like taking that muscle off the skull. So it's actually kind of beneficial to the skull to clean it off a little bit. If you were prepping a sample right now, you would just take a tissue, right? But if you were prepping a sample right now, you'd also scrape off some more of that before putting it in the collection. But yeah, it's something that we have to think about all the time. We never want to take something that is the last representative of a sample. But often, if you think of, we have these study skins, so they're kind of like these round little logs of what they used to be because the skeleton is removed and the skin is dried. So when I'm taking a sample for that, I can cut something as small as 1 millimeter by 2 millimeters and get adequate genomic data. From oftentimes I will take a skin clip from along the suture line where the animal was actually dissected for preparation. So it's not something necessarily super important. We try to leave every really important features on the skull. We don't want to really take bone and digest it for DNA unless we have to, because that has more morphological signal. So yeah, it is something we just constantly have to weigh the options. As a genomicist, it's pretty easy for me to see the value of getting DNA out of these specimens. But there was a time that a lot of curators before me did not want destructive analysis done. So it's just something we weigh on a case-by-case basis.
Jim?
Timothy Thomas, I'm a member of the Society. I want to ask a question about the over-preparation, which I might say, of the museum specimens. Once I went to the museum to look at a type specimen of a fossil turtle. I was very excited because, I was getting to see the type specimen. They were very nice at the museum, very nice, very helpful. However, the specimen was so over-prepared, you could barely tell what it was. They had, and I'm thinking about your domestics and if you think of them as preserving the skull, or you can think about them destroying the flesh, right?
Right.
It seems like with modern technology moving so quickly, keeping things out of the museum might contain more information when you get around to studying them.
Well, I'm not sure I understand what you mean keeping them out of the museum, but oftentimes now when we would collect a specimen or a series of specimens, you will do multiple types of preparations on them, not necessarily all made into dry study skins. For one, it takes a lot of time in the field to do that. For two, like you mentioned, you lose all the musculature, you lose the internal organs. So oftentimes now we will take, the first thing you would do, you euthanize the animal, and you take high quality tissues, and then you alcohol fix or formalin fix the entire remaining carcass, and you can store it like in a fluid preparation. So that way you can then use a CT scanner or an X-ray machine, and you can get all kinds of information. You can sample gut contents. You can do isotopes on hair. So you can get a ton more information if you have some prepared certain ways and some prepared other ways. We've actually also found that you can go back to some of the dry skins and actually comb them for like fleas. It's like, so ectoparasites still attached to the skin. So you can get those much better from a dry skin than a fluid skin. So there's a lot of benefits to different types of preparation. But yeah, a lot of the things we do now would be more in the world of fluid preps with tissues taken before any formalin is introduced.
Yeah, like I think about the National Museum in Brazil, which burned to the ground, and they had all of that Amazonian flesh, right? And all burned. And there's something good about having a museum concentrate everything so a person like you can go through it all. On the other hand, you lose it all in one day. And, it's kind of a balance between in the museum and in multiple collections in different varieties around the world.
Yes. Yeah, I agree that it definitely was a big tragedy. And across our building, we do have all kinds of different fire doors that protect different parts of the collection. So if there was a fire in one area, hopefully it would be contained to a certain area. And all the cabinets we have now are metal. Pretty much, we don't have very many wood cases left in our collection, and other departments in the museum they do still. But as you can imagine, it's a very expensive endeavor to replace all the cabinetry. So we try to do our best to keep things as contained with, obviously we have sprinkler systems throughout the whole building and all these heavy fire doors, so that should something happen, it would not be completely catastrophic. So, yeah.
Do you have a program with the American Museum of Natural History or the British Museum to ensure that there's a distribution of specimens, like what they're missing that you have duplicates of, they get one? And
we don't have necessarily a program that I'm aware of, but there is a lot of redundancy between the collections. Maybe different locations, but certainly a lot of taxonomic overlap.
So yes, we have a question here.
Zamankova, thank you very much for a fascinating talk. I'm a member. So you have been talking about million years ago and the effects on speciation and so on. Now, I am worried or concerned about or curious about more recent issues such as Suez Canal, Panama Canal, raising water levels. Is somebody actually preparing to study the effects of these things on diversity, speciation, etc.?
Yeah, I think there's a lot of modern ecologists. I mean, we know we're in the 6th mass extinction right now. The rates have never been higher in global documented history. It's a kind of a tricky thing because some of the things that I aim to study, I go into the cabinets and they've been there for 150 years and they're still not described taxa. So I try to work on that realm of highlighting where biodiversity is, because if we don't know, like this lemur, I had no idea that there could potentially be a cryptic species of a very large diurnal lemur. If we don't know something is unique, it's not necessarily going to be protected out of people's goodwill, right? So we need to have these taxonomic decisions kind of made by scientists to help inform current conservation, which can work as kind of a conservation umbrella for animals in the similar habitat or ecosystem. As someone that doesn't necessarily study exact human climate change and anthropogenic effects on speciation loss, I don't have much better of an answer, I don't think, to your question. Oh, I'm sure there's many more people working on that than there are people working in museums like me.
So I have a kind of technical question about the integrity or lack of integrity of the things that you're actually trying to sequence, the RNA and the DNA and the viruses. So if you taken like EMs and looked at things in a non-destructive way as possible to see, what is actually there as opposed to, what these isolation methods are able to recover, and also whether there's any intact virus, which, depending on your prep method, you might be able to see in an EM.
Yeah, so when we sequence DNA, you can actually look for patterns of degradation. It's called cytosine deamination. So you actually will get uracil to cytosine changes in nucleotides, and it makes this very distinct pattern. So the older your sample, the more likely you're going to see these deamination patterns. So that's one way to look and tell if your sample's authentic. So people that study woolly mammoths and other really ancient species are pretty much expected to show that they found these deamination patterns as a way of validating that it's not contamination. Obviously, a long time ago, there were these papers in science that claimed to have sequenced DNA from dinosaurs, right? And those were found to be all contamination. So these are kind of ways that we now kind of go about ensuring the authenticity. I've talked to someone at Fort Detrick, actually, that was going to look and do, what is the protein method for SARS-CoV-2? He was going to make slides, histological slides for me to confirm anything we found was intact SARS-CoV-2. But that would require him to make a histology prep. Was it fish in situ stuff I never do with cell biology, so I can't remember off the top of my head.
Do you know what he's talking about, Carl? Fish in situ to see if there's intact virus. I think we're talking past each other. Could you make a few comments about the conservation efforts in Madagascar for these species? Is there any success at all? Any hope?
Yeah, conservation in Madagascar is a very, very sad situation. I think it's estimated that somewhere between 50 to 80% of the forest cover is gone in Madagascar. Part of that is from the natural aridization of Madagascar. It kind of sounds like we have some rodents chewing on our lines in here. They know that I am a friend, not a foe.
And make a speech.
But yeah, there's a ton of groups. Everybody and their brother is a primatologist in Madagascar trying to save lemurs in Madagascar. And there's been a lot of strides made. I have no idea what COVID has done to that because their main problem in Madagascar is enforcement of national park boundaries. There's tons of poaching. It is incredibly, incredibly poverty stricken. I think it's like one of the worst countries in the world in terms of poverty. And when you go there and you see people that, you know, how they live and how they actually have nothing but the little tiny village where they can go into the forest and hunt a lemur, you know, a small 3 to 4 kilo lemur is a really big meal to them. So it's easy here to criticize people for poaching, and it's less easy there when you see what they're actually living with and on, no access to education and healthcare. So all these sorts of things kind of confound in making the poor stay poor, right? And without giving a huge influx of aid to Madagascar, I think that the situation is going to continue to be dire. I actually am very worried of what's happened since COVID because of just, you know, people, external researchers and NGOs not going there. I don't know what's happened. And, you know, there's so much unique species there. I think, what is it, over 70% of all animals and plants in Madagascar are endemic. So it is just like a really special place. And when you go there and you see these forests intact, they are really incredible. So we just have to keep fighting the good fight and try to keep providing the data that will help gain dollars towards these efforts.
I have another question here in the front row. Bob?
I'm a member. Has your research shown any relation between the species and location where you studied it and some other somewhat related species?
I'm not sure I understand the question.
Are these species completely unique to your area, or you found that there might be some relation somewhere in the past to others?
So for the lemurs, we found a number of sites where we think that this cryptic lineage might be found from modern samples. We could go model the ecological niche based on historic climate data and see where they may have been distributed. That is something that I've thought about doing for that project. I don't have any other good examples of the things I've presented where that might be as strong of a case as with the Shifox.
All this work is based on animals, mammals in this case. But while you were talking, it reminded me of Richard Dawkins' book, The Selfish Gene. And rather than, his argument would be that we're all just vesicles carrying the genes. And the real entity is the gene itself. So the question is, if you picked out a specific gene and then watched how it changed in the same situation, the big advantage there is that here you're doing qualitative work. you can describe this or that. Whereas there, you could begin to do quantitative work because you could see the rate at which it's changing and the mutations and that sort of thing. I've seen that done for the spike protein for COVID-19 and also for the beta-lactamase gene for antibiotic resistance. And they're very dramatic, and you can really see how the survival curve is not curves, it's actually the landscape changes. And I don't, has anybody thought about doing that for a specific gene? One of the things that intrigued me with the lemur was that they have variations in their ability to see color. And the
Right,
ones that
yeah.
are high up in the tree that need to pick fruit have the red, the capacity to see red, whereas the others don't.
Yeah, I actually worked on a paper where we looked at opsin gene variation in across lemurs. In this group of lemurs, there's not any variation. They're all completely diurnal, and they all see the same amount of shortwave opsin genes. So, but yeah, that's an interesting approach. I'm a big fan of Richard Dawkins. So, I've always just been such a mammal-oriented person. I've never thought of just dissecting a gene, but if I did that, I would just study viruses instead, I think.
So here's another question about what you said earlier about Madagascar. So you mentioned that it's this cultural practice where if you burn land, you own it. So you also showed a map of how much the forest has gone down since 1950, I think, with the implication that this is a pretty recent, devastating environmental impact. So if this cultural practice has been around for presumably a long time, why is this just now becoming such a problem?
Yeah, that's a really good point. Madagascar is actually one of the latest islands to be occupied. Despite its large size and proximity to Africa, the first colonizers were actually from Southeast Asia. Bizarre. You can see that today in the language. So there's a lot of similarities to Malagasy is a lot similar to Malay, which is very interesting. I go there and I switch languages between when I try to say hi to people, because they're so similar. But one of the main problems is overpopulation. So historically, yes, when they had small colonies around the edges of Madagascar, they would have slash and burn agriculture, and it wouldn't be that big of a deal because there wasn't, ubiquitous human occupation of the island. But since I can't remember the date, but I think it's something only as little as the past 6,000 or 8,000 years, Madagascar's been occupied by humans. I think that date changes based on evidence in the anthropological record, but it's very short. Unless there's been long times of, either lack of fossil evidence and things like that. And I'm not an anthropologist, so I don't have the most up-to-date information there. But I know it's very recent in terms of, especially being so close to Africa, the cradle of humanity. So I think part of it is just simply, people having a huge birth rate in Madagascar and having really a lot of kids, and then the population growing across the island compared to, and in conjunction with having, a shorter history on the island than might have been expected.
Any other questions? Anything from the web? Oh, Tim has another question.
No, I just want to say that my daughter has a program in Madagascar, in Manambo Special Reserve, if you're familiar with it.
Yeah.
It's in the south. Are you familiar with it?
I have heard of it. I haven't been there. I've been to about 6 locations in Madagascar, but
It's in pretty good shape, but if you look at the satellite pictures, the devastation from farming, and the big problem in Madagascar is food insufficiency.
Right.
And that's the terrible. She also has a program in Borneo, in Gunung Palung National Park.
Okay.
That's in the Indonesian side.
Yes,
and it's a lowland
yes.
rainforest.
Sounds like I should talk to your daughter.
Yeah, we should. I'll give you. I'll get you connected with her. But people are working.
Yes.
But it is
It's
difficult.
very difficult, yeah.
Yes.
And before going to these places, it's very easy from the outside in to, shake your fist at these people and say, well, why don't they just stop poaching? And don't they realize how special lemurs are? No, they don't, because they have never left their village in their life. They've always seen these lemurs, and to them, it's like us seeing a raccoon. you know? So like, they don't understand how important and regionally endemic these animals are. So, you know, once you get there and you see what they have and what they don't have, it makes it a little bit, you have a lot more compassion, at least I
Right.
did.
For example, they don't have bicycles.
Right. It's a commodity to get a bicycle. Yeah. So the
Yeah.
program I worked with is called Madagascar Biodiversity Partnership. And they have been there, again, like I said, for 20 years. They have all kinds of community programs and a huge reforestation program. So they're actually paying people to go out and plant thousands of trees every week. And it's been amazing. You know, I went and saw some plots they had planted 5 years earlier, and the trees are already, you know, 4 or 5 meters tall, because things will grow fast when you give them a chance and, you know, stop making farmland. And, you know, we talked about doing a lot of projects for monitoring those patches, because, you know, the first thing to go is some of the bigger lemurs like the shafaks from these forest fragments. Well, once they have grown for a couple years, you start seeing the little cute, adorable mouse lemurs. They come back quick because they eat really tiny things and they're easy to have at high density. And then, you know, you start to see the cascade and, you know, the improvement of the forest. So yeah, there's a lot of efforts for reforestation across Madagascar. And people are trying, but, you know, it's a lot easier to burn a field than to grow new trees. It's not as easy to replace.
Yeah, his daughter Kinnery, she's got an amazing program. And she has a book too, which is, I forget,
Okay.
yeah, she's really be worth beating.
Yeah.
A Force of
definitely.
Nature. Are there any more questions? If not, thank you, Melissa, for generously sharing your time, for putting up with all our equipment failures, despite our pre-testing. Before you go, I'd like to give you a small token of appreciation, which is a copy of the first volume of,
Thank you.
volume 1 of the PSW Bulletin, covering the early period in 1871 and a few years after. It explains why the society was founded, who founded it, and why they called it the Philosophical Society of Washington.
Thank
it
you
means
so much.
love of knowledge, but and then there's some interesting short minutes of early meetings, and including a description of a presentation on the calculating the value of pi.
Great.
The 30 decimal places was a very big deal back then. Now we do an infinite number of decimal places. So there's such a thing as progress.
Great.
All right. Thank you very much.
Thank you so much.
Before we go, a few closing remarks. Our next talk by Kim Prather will be lecture number 2456 on March 25th. Kim is the Distinguished Professor and Chair in Atmospheric Chemistry in the Department of Chemistry and Biochemistry, and Director of the NSF Center for Aerosol Impacts on Chemistry of the Environment at the Scripps Institution of Oceanography and the University of California, San Diego. And she'll be speaking on ocean microbes, aerosols, climate and health, tiny critters, huge impacts. This lecture may be held via Zoom. I'm waiting to hear from Kim whether she can travel here. If she's not able to, then we will have it by Zoom. Please watch the website for further information. Final word on this will also be highlighted in the announcements that will be sent next week. The next lecture after Kim has yet to be scheduled, so keep an eye on the website for that. And then on April 22nd, we'll have our 2458th meeting, and the speaker will be Elizabeth Strychowski. She's the group leader of the Cellular Engineering Group at the National Institutes of Standards and Technology, and she will be speaking on measurement tools made from biological parts and on efforts to define the minimal genetic complement required for normal cell functioning. There are several recent publications on this topic. Lecture number 2459 will be held on May 6th. The speaker will be Rahul Saini. He is the CEO at Teleatry. He's the MEMS director at NanoRetina. He's the director of micro and biosystems at Zyvex Labs, and he's also the chief technology officer in NXT STEM. I have no idea how he's going to find time to come here and give a talk, but he said he would. He'll be speaking about implantable electronic devices set to revolutionize certain aspects of medical care, illustrated by an implantable retina that's now in clinical trials, and an implantable vagus nerve stimulator that is also in trials to control epileptic seizures. And finally, on May 20th, we have lecture 2460. The speaker will be Bob Bellaram of JPL Caltech, where he is the chief engineer of Ingenuity. And of course, he'll be speaking on Ingenuity, the first flying machine on Mars. Please check the PSW website often for up-to-date information on meetings and whether lectures will be in person or via Zoom. COVID and COVID-related public health measures are constantly changing, and PSW is adjusting plans accordingly. So look on the website to know exactly what we need to do. I hope you'll all join us for these meetings. And one last thing before we go, let's thank tonight's volunteers for producing this meeting. Robin Tiller, Bill Goodwin back there at the streaming table, James Heelan, who read the minutes, Ann McQueen, Jared McQueen, Robert Thompson, Noah Block, Cameo Lance, Laurel Kane, and some friends. And with that, I will now adjourn the 2455th meeting of the Society. I wish everybody a good morning, afternoon, evening, or night, wherever you are. The meeting is adjourned.